Hi, I'm MJ and I'm a professional research assistant at the University of Colorado Anschutz Medical Campus, and I like to hike in my spare time. So today I'm going to teach you about DNA and about polymerase chain reaction. So DNA is in all of us, and it makes us who we are. And what it is it's a double helix molecule. And what we can do is we can actually study it by making a bunch of copies of it. So what we can do is we put our DNA sample into a tube with primers, which will target whatever we want to look at. And we can put it into a PCR machine, a polymerase chain reaction machine. And so what happens is, as your sample heats up, your double helix will actually melt and denature, and both of those strands will come apart. And then we can actually make a copy of it and get a whole bunch of copies so that we can actually visualize it on a gel. Okay. So what I have here is an agarose gel, and in this gel there is a dye called ethidium bromide that binds to DNA and allows us to visualize it on the gel. Take our gel, put it in our gel box, and this gel box, what it does is it runs a current through the gel. And this works because DNA has an overall negative charge. So as this current runs, the DNA, because it's negatively charged, will be pulled towards the positive electrode based on how big it is. So the smaller fragments will move a lot further down the gel, and the larger fragments won't move quite as far. And for everything to work, we need a buffer for our electrodes, and we just need to cover the gel completely. And then we can go ahead and start loading our DNA. So you always want to run a DNA ladder, and I actually have a picture of it right here. This helps you figure out what size your DNA fragments are that you're interested in. So that's what you always want to put first. So that's what I'm going to do. Okay. I'm always a little shaky, so I hold my hand here, and you just dispense it right into the wall. This is my amplified DNA from my PCR, and because we want our sample to sink down into each of these wells, we need to put a dye, and this is just something that makes the DNA a bit heavier so it'll actually sink down. Okay, and so I'm going to mix my sample with that. And then I'm going to load it right next to the ladder. And you can see it kind of just sinks down into the well there. And that's it. Okay. Alright, so our gel is all done. So you can kind of see the dye has run down the gel, and that kind of gives you an indication that it has actually worked. Now we're going to take it over to the UV light to look at our DNA bands. So because it's UV light, you'll want to shield yourself from that, and we'll go ahead and turn it on. Okay, and you can see right here is our ladder that we loaded, and here is all of our DNA bands. So our PCR looks like it worked. and you can tell the size of your band based on this ladder, and it looks like these bands are a little bit over 300 base pairs in length.